Okay, we'll keep moving forward as we talk about the rest of the biogenic amines, right? So these are non-catecholamines now. These are the two other classes of biogenic amines. And the first one is the neurotransmitter histamine. The synthesis of histamine is from a single histidine amino acid. So if we take the histidine, and in one step, histidine is converted into histamine. And that step is detailed over here to the right. Pretty simple decarboxylase enzyme gives us the histamine neurotransmitter. Transport, right? As we load histamine into vesicles, that occurs via the same VMAT as the other catecholamines. So loading the vesicular monoamine transporter. Mechanisms of reuptake aren't well understood. So for reuptake, just put unknown. Breakdown. We've seen it before, but it's the same MAO, the same monoamine oxidase. And there's a specific enzyme. You don't need to know the name of the specific enzyme, but there is a what's called a histamine methyltransferase. So there is a specific enzyme for histamine. With respect to the receptor, there are three types of receptors. So three different receptors, and they're all G protein coupled. So none that are going to open ion channels. You might have heard of histamines because you've taken an antihistamine, right? Antihistamines are able to cross the blood-brain barrier to interfere with the role of histidine. Whoa. And so one of the effects of histidine, one of its functions, sorry about that, one of the functions of histamine is to mediate arousal and attention. So a similar function to other neurotransmitters we've seen like acetylcholine and norepinephrine. And so it would make sense that if antihistamines are able to cross the blood-brain barrier to interfere with one of these G protein coupled receptors, that we would see sleepiness, tiredness, right? So reduction in arousal and attention. If we were to block this, right, we get groggy, we get tired. There's been also some discussions that because of the cells that release histamine and the cells that are around the cells that release histamine, that we think that there's some potential for histamine to be able to regulate blood flow in the brain. So that's about it for histamine. We'll move on. We'll talk about serotonin next. Serotonin, the last of the biogenic amines, is catalyzed from the amino acid tryptophan. So the synthesis. In a couple of steps, tryptophan, the same amino acid that people think makes you tired at Thanksgiving, right, gets converted to one. There's an intermediary here. The second step converts it to serotonin. often called 5-HT because of the chemical that it's made out of, 5-hydroxytryptamine, but serotonin is certainly good for our purposes. Packaging, right? Loading into neurotransmitter vesicles. The same VMAT that we've seen before. Interestingly, though, the transporter to take it up is unique. It's called SERT. So if we're taking it up into the presynaptic neuron again, so if we talk about reuptake, so SERT, this serotonin transporter, right, is unique among the biogenic amines with respect to uptake. And the most common antidepressants that we use now, like Prozac, inhibit SERT. So drugs like Prozac that are known as SSRIs, selective serotonin reuptake inhibitors, block SERT. Okay. With respect to breaking it down, once it gets into the cell, it's broken down by MAOs, monoamine oxidases. All right, so we'll say breakdown. MAOs. So if you consider what makes a good antidepressant, we want specificity, right? And I think as we've seen, multiple neurotransmitters are broken down by MAOs. So if one takes a MAOI, a monoamine oxidase inhibitor, to treat depression, you're not just going to treat it by prolonging the presence of one particular neurotransmitter, but multiple at any synapse, right? Whereas SSRIs give us the ability to specifically tinker with the serotonin system. So most receptors for serotonin, and there are many, most of them are G-protein coupled, GPCRs, G-protein coupled receptors, okay? And the effects that they mediate are many as well. Whoops. Go back. The effects are widespread from emotional behavior to motor function, to satiety levels, right? The feeling of hunger. And so it's common, right, for eating disorders for folks to take serotonergic drugs because they regulate satiety. So if someone has an eating disorder, it's this system that's thought to be involved. So we can talk about drugs for eating disorders. Those drugs are given and they target these G-protein coupled receptors that use serotonin as their ligand, okay? There is one group of ionotropic receptors, right? I think the book says there's something like 20 different serotonergic receptors. There is one group of ionotropic receptors within that. And in this case, when serotonin binds to these receptors, they open up non-specific cation channels. Of course, what this will do is depolarize the cell, so these are considered excitatory actions. Okay. Other neurotransmitters, besides all the ones that we've talked about, all the biogenic amines, the GABA, the glycine, the glutamate, right? We've talked about many. Acetylcholine is in there. We should talk now about purinergic neurotransmitters. So when vesicles release their neurotransmitter, all vesicles contain ATP. And so it turns out that ATP works as a co-transmitter. So in addition to being used, the energy currency of the cell, so to speak, that ATP can work as a neurotransmitter. Now, depending on the case, once ATP is released, there are sometimes enzymes that are tasked with breaking down ATP into their adenosine portion. Right, so that's all ATP is, right, is the adenosine, this nitrogenous base with 3 phosphate groups attached, right? Well, when enzymes can attack ATP, basically get rid of the phosphates, we're left with adenosine. And adenosine can also work as a neurotransmitter, though not in the classic case, right? The adenosine is not a classic neurotransmitter. Why? Because it's not in vesicles, right? It's not packaged into vesicles. Nor is its release calcium dependent, as all of these other neurotransmitters we've been talking about are. So not calcium dependent release. But it can still function as a neurotransmitter. So let's talk about the receptors. These receptors are known as P2X receptors. They are ionotropic. And so the purine, the adenosine is the reason we get the term purinergic is adenosine is a type of purine, right? You might have heard the term purine when you think of structure of DNA, right? A's and T's and C's and G's. This adenosine also falls into the category of a purine. So a purinergic neurotransmitter like adenosine or ATP has a P2X, that's a purinergic receptor. These things are ionotropic, that is, when they open, they allow ions in, of course, and they're heterotrimers. So that means that they're made up of 3 different subunits, right? And if we were to look top down, oops, that's a poor drawing. If we were to look top down, 1, 2, 3, there's a pore right in the center. That's still a pretty poor drawing. I'll show you on the picture in the upper right. Okay, there's the pore. And you can see, right, this line is indicating here in this picture where the ATP or the purine would bind. And here's one subunit, right, of the 3 subunits. And allegedly it looks like a dolphin, so people talk about that. These are non-specific cation channels as well. Specific. So they are excitatory, generally. Now, we know they play a role in pain from a functional standpoint. But despite that, we're not certain what their role is in other cells. We know these receptors are found in other cells, but we're not sure of their function. There are a couple of other receptors besides these P2X receptors. So 2 other receptors. One type that prefers the adenosine, and a second type that prefers the ATP ligand. It's interesting, the one that prefers the adenosine ligand is inhibited by caffeine. Right, so this blocks this one type of receptor, right? So it inhibits the adenosine signaling. So we think that some of the stimulatory effect is by blocking this adenosine receptor. Now, like I mentioned before, these other 2 types of receptors, we know where they are. They're in the brain, they're in the heart, they're in the kidney, but we don't have a particularly good idea of how they function. Okay, that's 5 videos in, right? If you think they're all about 15 minutes, that's about 75 minutes of content, which is roughly what we would do in a normal class time. So we'll call this a day, right? Again, you can look forward to 4 to 5 of these videos per class session that we meet. Let me know if you have questions. Let me know if you want to do virtual office hours. I'm welcome for that. Otherwise, I'll have more of these videos up for you on Wednesday, and I will talk to you then.